Next, I would like to introduce Dr. Jennifer Maier. She's currently a postdoctoral research fellow at the University of California, Los Angeles. And she'll be speaking to us today about new approaches to disease modeling, developing new non-model model systems. Welcome.
Thank you. Okay, so hi. Thank you for having me here to speak today. This has been a great meeting so far, and I'm really excited to share some of the work that our lab has done with Monodelphis domestica, the gray short-tailed opossum, some stuff that we've done in the past, and then stuff that we are doing right now. So Monodelphis domestica is a marsupial mammal, just like kangaroos and koalas. They're a group of mammals that are native to the Australias and the Americas, and so I'll give you a little bit of an intro about this model system because not everyone is really that familiar with it, and then talk about some of our work. So whenever I say that I work with opossums, a lot of people cringe and have this horrified look on their face because they're thinking about Didelphis virginiana, or the Virginia opossum. So this is the black and white one that you'll see walking around or roadkill on the side of the road. So this has also been used as a model in biomedical research. It's not as commonly used as, say, our species, but they have been used in the past. So our species is native to South America. It's a lot smaller than the Virginia opossum, with males getting up to about 150 grams at the most. And they're a lot more docile and amenable to laboratory study than the other species, to put it very nicely. They're also a pouchless marsupial. So if you look right on this belly region, this is a female opossum with a litter of newborn pups, which look like small pink jelly beans. So unlike some of the more common marsupials that do have their young covered in a pouch, these ones, they're exposed right on the belly. They're fairly straightforward to raise in a laboratory setting. People have bred them for quite a long time now. One advantage is you can breed them year-round. We have noticed seasonal differences in our colony, at least, where they are less successful with breeding in the winter. And as we've just moved from Illinois to California, I'm wondering if that's going to persist. So excited to find that out. Is there a way to make the video play? No. Okay, so this is an example of our typical setup. So we house our animals in rat cages. This is a female with roughly 3-week-old pups stuck to her belly. And so they'll stay on very easily, and she has no problem moving around with them at all. They don't seem to bother her, so she's caring about her day, eating, drinking, and burrowing. And they'll just hang right on. And then eventually when they get a little bit older, they can come off and walk around and eat, and you'll see them clinging to her back when you come in sometimes. But they're just housed in a standard rat cage with some bedding and nesting material. We give them a small crock of food, and they have a water bottle, because unfortunately we discovered that they do not comprehend how to use the automatic ordering systems that you would normally give to mice. And then they have a little, just a standard mason jar like you would put spaghetti sauce in, that they build their nests in. So their litters can range from 2 to 13 pups. They can also have more embryos, but they don't have enough nipples in order to feed every single embryo that's born. So what's interesting about them is when they're born, they crawl to the nipples, and then they actually fuse there, so it's very hard to remove them for several days. And being marsupials, they have a very abbreviated gestation, so their gestation is only about 14.5 to 15 days, and then the young are born. So there's some considerations to think about if you want to do work with opossums. One is that they are a USDA-regulated species, so you'll have a few additional things to keep in mind in terms of records and procedures, in addition to your institution's IACUC and other regulatory boards. They're also a non-social species, so you cannot group house them or they will fight to the death in some cases. So the only reason that we ever put our animals together is if we are breeding them, or if they are very young weaned animals, we'll group house them for a few months. But if they start to fight, obviously we have to separate them. They can be fed on a commercial opossum chow that's now become more widely available. We used to feed ours basically science diet cat food, and they will eat it, but they definitely like the stuff that's for them more, and then we supplement with insects like mealworms and crickets and fruit. They are a lot slower to mature compared to a mouse. While they only have a 2-week gestation period, it takes 2 months before they wean from their mothers, and then 8 months to reach sexual maturity. However, they can live up to 3 years in captivity. They do lose some fertility. And then, of course, if you're breeding any kind of animals, you also have to try to maintain genetic diversity. So why work with Monodelphis? So on the left, this is a newborn mouse. This is a newborn opossum. There's some pretty big differences in them. The opossums, because they have such an early birth and short gestation, it's commonly reported that they're equivalent to about a 10.5 to 12.5 day old mouse embryo, depending on the organ system you're looking at. So because of their short gestation, a lot of their systems are only rudimentarily developed. They also have a unique biology compared to placental mammals. They have a slower metabolic rate, a lower body temperature, and sometimes this will give them advantages over a mouse or other placental model. They also have some human orthologs that are relevant to human diseases that you can't always find in the mouse. So it might be that this is a better system in some cases to study certain diseases. We have used them in comparative studies comparing placental mammals versus marsupial mammals because as a marsupial, it has only a rudimentary placenta. It doesn't have a highly invasive one like a placental mammal will. And they also have this interesting disparity in forelimb versus hind limb formation, which it's a little hard to see here, but I'll talk about that more in the next slide. Oh, or not. So there are a number of resources and methods that have been developed for this species over the decades. They do have a genome available, which has been really helpful for a lot of the work that our lab does. We're mainly an evo-devo lab, so we look at a lot of different mammal species in order to ask our questions. The embryology of these animals is pretty well established from fertilization to just before birth, so there's a great staging guide available if you need to get timed embryos for various stages of limb development or craniofacial development. It's very easy to breed them in a laboratory. You can now do RNA-seq for these animals, so we've generated transcriptome libraries for the limbs. Other groups have generated them for other parts of the embryos. The opossum community is growing. There's a opossum base online resource that gives you a list of many of the researchers around the world that work on these animals and then what they do. There's also some genetic resources and other basic protocol resources available on this website. And a lot of techniques that you would do in mouse, you can usually adapt to opossums, one being in situ hybridization, so looking at the mRNA expression in the embryo. You can also do immunohistochemistry, immunofluorescence. It can be a little challenging to find antibodies that will work on opossum because, of course, most antibodies available commercially are not designed with opossums in mind, so sometimes you have to search around before you find something that's compatible. You can also do siRNA for a brief time in organ and tissue culture, and it's possible to do cell culture in these animals. So one of the things that our lab looks at is limb development and evolution. And the reason we look at the limbs is because, number one, they're very morphologically diverse. So in mammals, you have the bat, which is the only mammal that's capable of powered flight. You have dolphins, which of course have flippers and swim. We have our own 5-digit hand, and then we have an animal like the horse, which runs around on only 1 digit. And it's thought that this limb diversity of mammals has allowed them to exploit many of the habitats on Earth. Another reason we look at the limb is because it's already a very well-established study system, especially in animals like mouse and chicken. So a lot of the genes that are required to pattern and grow out the limb are pretty well known, and they are conserved in opossum. And another thing is that one of the very common human birth defects are congenital limb malformations, or CLMs. These can be caused by genetic mutations, and they can also be caused by something in the environment. So In order to learn more about how these are caused and potentially how they can be either treated or prevented, it's important to study the limb to find out how they're actually caused. So the limb basically starts as an outgrowth on the flank of the body. It becomes a small ridge that grows out from the side, and then it becomes a bud. So this would be an example of a limb bud. It's about as wide as it is long. There's two major signaling centers that are in the limb. This might be a review from anyone who's taken an embryology class. You have the ZPA, or zone of polarizing activity, which is on the posterior end. It secretes sonic hedgehog. And then you have the apical ectodermal ridge, which is along the edge, and it secretes fibroblast growth factors. There's a number of other players in limb formation, including the HOX-A and HOX-D family, gremlin, glies, and bone morphogenetic proteins. And so interaction of all of these genes is required to properly form and pattern and outgrow the limb. So if you have a mutation or something in the environment that's causing a mutation, you can have defects in this outgrowth and patterning. One interesting thing that we use the opossums for is their disparity in the forelimb versus the hind limb development. So this is an opossum embryo that's about a day or so away from birth. The forelimb is really well developed, and it needs these well-developed forelimbs in order to climb from the mother's birth canal to the teats in order for it to finish developing. And if it can't do this, it's going to fall off and it won't be able to continue developing. In contrast, the hind limb is still at a paddle stage, and so it hangs just kind of limply as they do this crawl, and then you see a lot of this development outside of the womb. And before I jump into this, we've also found that you can treat opossums with different teratogens in order to induce congenital limb malformations. So this is work from a former grad student in our lab. She used retinoic acid, and retinoic acid is a derivative of vitamin A. It's required for normal limb formation, but if you have too much of it, you can cause congenital limb malformations in most vertebrates, including frogs and mice. A common route of human exposure is usually through pharmaceuticals. For example, some acne medications that contain retinoic acid. And we wanted to ask, first of all, can you get CLMs by treating opossum embryos with retinoic acid? And then secondly, what is the molecular and cellular basis of these CLMs? So what's actually happening when we give them this? So we treated pregnant dams with retinoic acid orally, and then we collected the embryos before birth. And what we found was very highly penetrant phenotype. So all animals in treated litters were affected when they were given retinoic acid. So this is our wild type control litter. We just gave them the vehicle that retinoic acid was dissolved in. All of our treated animals had oligodactyly, so they were missing digits. They only have 3 here, whereas the wild type has 5. They also all had some sort of craniofacial defects, and a subset of them actually had encephaly. So we found that, yes, retinoic acid will produce not only limb but also craniofacial defects in the opossums. We also looked at gene expression. So we found that we looked at the AER and the ZPA, those 2 major signaling centers I told you about. So we looked at FGF8 in the AER. This is in a control animal. And FGF8, as you would expect, is expressed throughout the AER. Whereas in our treated animals, the expression domain of the AER is much reduced, suggesting that there's something going wrong with FGF signaling in these animals. I didn't show it here, but we also had a disruption of sonic in the ZPA. So the expression domain was a little bit smaller in our treated animals. We also looked at cell proliferation and cell death in the limb bud of these treated animals. And we found that throughout the limb mesenchyme, you have a decrease in the number of proliferating cells compared to control. And then if you look at cell death, you have a lot more cell death in the treated animals compared to control, especially in the region of the ZPA, which is one of the major signaling centers. So we concluded that if you administer exogenous retinoic acid to the opossum, you can get craniofacial and limb defects. This happens by disrupting the AER and ZPA, leading to a decrease in proliferation and increase in cell death. And interestingly, they seem to have higher penetrance to this than does the mouse. We also have looked at Thalidomide Teratogenesis, which you probably remember hearing about in the late 50s, early 60s. It was prescribed to pregnant women for morning sickness. And unfortunately, some of the babies born to these women had severe defects in the craniofacial, other parts of the body, the limb, and vascular deformities, such as the children here that have very truncated limbs. So mouse and rat embryos were actually found not to be susceptible to teratogenesis. Non-human primates and rabbits are actually susceptible, but it's difficult to get a large number of embryos for study. It's actually thought to work by disrupting angiogenesis or blood vessel formation, and it's becoming relevant now because thalidomide is being used in some countries as a treatment for leprosy and multiple myeloma. So we wanted to first ask, are Monodelphis embryos susceptible to thalidomide, and if so, do they have any similarities to human phenotypes? So we injected pregnant dams and then took the embryos before birth and looked at the phenotypes. And we had a spectrum of phenotypes. So about a third of the animals in the litter were what we called quote-unquote normal, in that they had this nice pattern of blood vessels, what seemed to be a normal-looking head, properly patterned limbs. About half of the embryos in every litter were what we termed mildly deformed, and while they look outwardly pretty normal, they do have blood pooling throughout the body. And then we had more severe phenotypes, which comprised the rest of each litter. So these animals were typically a lot smaller than their litter mates. They had no really discernible major blood vessels. Instead, they had pooling in the ventral side of the body. Some had truncated limbs or improperly patterned limbs, and they also had craniofacial defects. These are some more of our severe phenotypes. So once again, you can see the dramatic pooling of blood. Some had an improperly formed heart, and then we had a couple that actually had 2 amniotic sacs, or 1 amniotic sac that was constricted. So we concluded that Monodelphis does at least have some of the human thalidomide phenotypes upon treatment. It does have a very high penetrance for the mild deformation, and it does seem to be disrupting angiogenesis, causing defects in the heart, in the blood vessels, the limb, and in the amniotic sac, and then also in craniofacial morphology. We're actually still working on characterizing these phenotypes more molecularly, looking at gene expression and cell death and proliferation in these animals. Some other work that our lab has done using these animals has been work on the mammalian middle ear formation and intervertebral disc formation. Interestingly, they don't actually have intervertebral discs when they're born. That forms within 10 days of birth. And then other groups have used them to study things as diverse as spinal regeneration, melanoma, hyperlipidemia, and a lot of other questions. So they seem to be a great model for studying biomedical and evolutionary questions. So our major challenge right now with working with this model is there's no way to do knockout or transgenic modifications of these, which complicates functional testing. Last year we received a grant from the NIH in order to try and generate methods to make a transgenic opossum, and we're proposing to do this by using spermatogonial stem cell transplantation. So this method has been used in mouse and rats in order to do genetic modification. And a very oversimplified flow chart. We first take cells from the testes and we culture them differentially on different substrates. This is because the spermatogonial stem cells are only a tiny fraction of total cells in the testes, so we need to purify them out. So we plate them first on plastic, then collagen, and then laminin. That will get us our mostly pure population of SSCs. We'll plate them again on gelatin in order to get any last somatic cells out. And then we expand them on opossum embryonic fibroblasts that we have to generate from embryos. So we are at this point now and trying to refine our cell culture methods in order to get much larger amounts of these SSCs. And then eventually we hope to modify them in vitro, transplant them into a recipient male's testes, and then get our carrier offspring. So these cells have also been cultured from cow, sheep, and pigs in vitro. Our preliminary results, it seems like we can culture these cells from the opossum testes using very similar methods. We have gotten cells that have a expression profile consistent with SSC identity, and we're trying to refine our culture methods now before we take it further. And in the future, we'd really like to do CRISPR-Cas9 and transplant them into the opossum to look at different gene expression. So in conclusion, we really think that opossums are a great model for biomedical and evolutionary questions. They are amenable to laboratory study, and there's a lot of resources available for them with more under development. And it's our hope that once we're able to do genetic modification, this could probably give us some more insight into human disease. So with that, I want to acknowledge our funding from the NIH, my PI, Karen Sears, and then current and former members of the lab that have contributed to this work. Our former facilities at the University of Illinois that we did all of this work, and then our current ones at UCLA. So thank you.
Thank you very much, Dr. Maier.